Hi, I'm Dmitry Rodionov. My team present two posters at this conference, first by Stanislav Yablokov and second by Semyon Lein and Marat Kazanov, both describing phenotypic characterization of complex microbial communities. This novel approach allowed to combine genomic-based reconstruction of metabolic pathways for reference genomes with metagenomic profiling. It allows in silico prediction of major metabolic capabilities of microbial communities, such as nutrient requirements, utilization of different sugars. It uses binary phenotype concept, where 1 and 0 stands for the presence and absence of particular metabolic capability, encoded by complete pathway variant in the genome, and combine them into the binary phenotype matrix that captures predicted capabilities for hundreds of metabolites and thousands of microbial genomes. We developed the phenotype profiling pipeline, which contains three major steps. First, BPM construction for a subset of 2600 human gut microbiome genomes. Second, BPM expansion to a larger set of reference genomes, and we developed Phenotype Predictor tool in order to expand the BPM. And the third is BPM usage. We developed Phenotype Profiler tool, which use a taxonomic profile obtained from 16S or WGS metagenomic samples in order to calculate community phenotype index and phenotype alpha diversity and beta diversity metrics describing phenotypic capabilities of real metagenomic samples. To enable phenotype profiling pipeline, we first construct the binary phenotype matrix using reconstruction of metabolic subsystems or pathways in MCC platform, utilizing genome context analysis techniques such as chromosomal clustering, co-regulation of genes, co-occurrence of genes in the profiles. And we curate functional roles for enzymes, transporters, regulators, and assign pathway variants to each pathway and binary phenotype at the end, which form the binary phenotype matrix. Currently, the BPM include 2600 human gut microbiome genomes. Here is an example of sugar utilization pathway, where we specifically predict and curate metabolite uptake transporters in addition to catabolic enzymes. We curate different alternative pathways for sugar utilization and non-orthologous enzymes catalyzing the same reaction. We also annotate carbohydrate active enzymes involved in dietary fiber processing. Here is a fragment of obtained BPM for key human gut microbiome species, showing the variability of phenotypes for biosynthesis of vitamins, amino acids, utilization of sugars, short-chain fatty acid fermentation, and others across this key player of the microbiota. At the second stage of the pipeline, we use BPM to calculate several metrics describing phenotypic capabilities of real metagenomic samples. Input data are either 16S or shotgun reads, which are converted to taxonomic profiles by standard tools, and the Phenotype Profiler tool, which we developed, calculate community phenotype matrix, which is an average phenotype for each species in the taxonomic profile, and finally calculate three metrics: community phenotype index, which is a percentage of pathway holders, phenotypic alpha diversity, which is a diversity of alpha diversity of carriers of a particular phenotype, and also phenotypic beta diversity. For more details, please look the posters by Stanislav Yablokov. Since the number of isolated sequenced human gut microorganisms with available genomes is rapidly growing, and also uncultured bacteria with metagenome assembled genomes MAGs substantially expand the genetic diversity of these species, we developed a computational pipeline Phenotype Predictor for automated propagation of curated metabolic phenotypes from the reference BPM over the new microbial genomes. This Phenotype Predictor pipeline includes two major stages described further. In the first stage, we identified gene orthologs for each metabolic pathways or subsystem from the reference database in each novel genome using diamond-based gene annotation workflow. We specifically designed a new approach for functional annotation of multi-domain protein in this stage. In the second stage, we built predictive models for assignment of binary phenotypes to novel annotated genomes. We obtained phenotype rules by using the decision tree method combined with expert pathway rule curation, and we also selected 3 best performing machine learning approaches and trained machine learning models with either all features metabolic genes or subsets of features selected using forward feature selection method. Only a subset of subsystem genes or features was used in the predictive models, and this number of features varies depending on the pathway analyzed. Here we show example of obtained phenotype rules for butyrate production pathways, which are rather complicated and include 4 different pathway variants, P1 to P4, but the decision tree based phenotype rules for this pathway are relatively simple and involve only 6 different genes. Conclusions. Predictive phenotype profiling is a novel approach for comparative analysis and classification of metagenomic samples. Binary Phenotype Matrix encodes metabolic phenotypes inferred from expert curation and in silico genomic reconstruction of metabolic pathways across large collection of genomes. Current BPM captures phenotypes for utilization and production of over 100 metabolites and over thousands reference genomes. We developed computational pipeline for automated propagation of curated metabolic phenotypes from the reference BPM over new microbial genomes. The obtained formal phenotype rules and machine learning predictive models allow phenotype assignment with over 99% accuracy. We applied them to 2 recently appeared public sets of human gut microbiome genomes, totally 2300 novel genomes, to obtain an expanded BPM covering a larger phylogenetic space. We would like to thank Andrei Osterman from my institute and Pavel Novichkov from Phenotype startup for useful discussion and encouragement, and to the multiple students for help with curation of metabolic phenotype database in reference microbial genomes. For more details, please check our posters by Semyon Lein and Marat Kazanov and Stanislav Yablokov. Thank you for your attention.